My name is Peter Zerhouni. I'm the CEO of Index Pharmaceuticals. And we'll start with our standard disclaimer regarding forward-looking statements before we get into the actual presentation. So Index is a drug development company. We're based here in Stockholm, close to the campus of the Karolinska Institute, where also the research behind the company once originated. We focus on immunological diseases with high unmet medical need. And we have a very experienced board to support me and the management team, which is also quite experienced. And our foremost asset is called Cobitolimod, that we are developing for ulcerative colitis. And we've come a long way. We are currently running a phase 2b study with this drug, or drug candidate, which is a dose optimization study that we call CONDUCT. And the strategy is to partner Cobitolimod based on the data from this study ahead of phase 3. Apart from Cobitolimod, we have a broad portfolio of chemically similar assets. These are oligonucleotides, so short snippets of DNA-based single-stranded DNA-based oligonucleotides. We call them DNA-based immunomodulatory sequences, or DIMS, that has a broad potential in inflammatory diseases by modulating the immune system via toll-like receptor 9. And I'll talk to that in a minute. We are listed on the Nasdaq First North list here in Stockholm. We IPO'd in 2016, where we raised 250 million, primarily to pay for this phase 2b study. Our main shareholders are VCs. We have SEB Venture Capital with a bit more than 20%, Swedish Industrial Fund or Industrifonden at 19%, and Swiss-Norwegian Life Science VC Neomed Management with 10%. Together they hold 50.1% of the company. So they're still in charge. Ulcerative colitis then, it's a terrible disease with high unmet medical need. It's a chronic inflammation of the colon that leads to ulcers. And it comes and goes in flares, so active and inactive stages. So when you're in a flare, or when the patient is in a flare, they suffer from the typical symptoms of very frequent blood and mucus mixed loose stools, so basically bloody diarrhea. And they might have to go to the toilet up to 50 times per day. This has a very high negative impact on the quality of life. Patients get chained to their home, they can, or the bathroom even. Very difficult to entertain a social life, to uphold a job, etc. Typically this disease debuts or presents somewhere between 20 and 30 years of age. So a sensitive age when most people find a partner, settle down, you finish your studies, you start a family, etc., get your first job, and then it's very difficult. Then, of course, you have the disease for the rest of your life. It's a chronic disease, so you never get rid of it. And there are a number of drugs available to treat these patients, and some of them have been around for a long time and work okay in a milder patient population, but then we have a fairly sizable group that don't really respond to the currently available drugs, and particularly when they have a bit more difficult disease or moderate to severe ulcerative colitis, they don't have that many treatment alternatives. So let's look a bit closer at the moderate to severe ulcerative colitis treatment alternatives. So the leading drugs here are biologics. We have currently 2 classes of biologics for this patient group. The anti-TNF-alphas, so that's Remicade, Humira, Simponi. This is the world's best-selling drug class. Can be used in a lot of indications, but very important here in moderate to severe ulcerative colitis. Then we also have a fairly new class called anti-integrin antibodies. There's only one product approved, Entyvio from Takeda. But all of these suffer from similar weaknesses. They help a fairly small proportion of patients, so there is limited efficacy. And efficacy in this indication is expressed in terms of delta, meaning how large share of the patients in the active arm go into remission or become symptom-free, compared to how big share of the placebo-treated patients become symptom-free. And the difference between the two is the delta. The currently approved biologics have about a 10% delta, meaning if 10 out of 100 patients get symptom-free based on placebo, then an additional 10 patients, or in total 20 patients out of 100, become symptom-free by active treatment. Which means 80 out of 100 patients are still not symptom-free, and of the 20% that are symptom-free, actually only half of them became symptom-free because of the treatment, and half of them would have become symptom-free with just the placebo treatment or no treatment. So this is, of course, looking from the outside, I mean, that's a terrible record for a drug or for drugs that are associated with severe side effects. So these are not benign drugs. I mean, these are pretty tough treatments. And in addition, a lot of the primary responders, the ones that respond in a first course of treatment, about 50% of those stop responding to the drug within 1 to 2 years, meaning that then you need to find another alternative. Still, they sell for more than $5 billion per year. So, of course, you have to pay for all 100 patients, even if only 10 actually respond to the drug or get symptom-free based on treatment with the drug. So there's a large commercial opportunity that comes with this big unmet medical need, and that has attracted a lot of companies to develop assets for this indication. So there are a lot of drug candidates in development to treat moderate to severe ulcerative colitis. However, none has really cracked this issue of efficacy. So the deltas, or the efficacy, is still around 10 to 20%. So 10 to 20% of the patients that become symptom-free based on the treatments. We had recently the first JAK inhibitor approved. That's a new drug class to this indication. It's called Xeljanz from Pfizer. It's already approved in RA. They had a delta of 10% in one of their phase 3 studies and 13% in the other, so very similar to the currently approved drugs, or the previously approved drugs. And then there are two other mechanisms of action or classes of drugs in phase 3. We have anti-IL-23s, so those are monoclonal antibodies. Stelara from J&J is the one leading the pack there. And then we have S1P receptor modulators. Ulcerative from Celgene is in phase 3. But for each of those classes, there are several other drug candidates that's coming from behind in phase 2. Still, many of these are associated with increased risks of severe side effects. So these are still tough drugs. If you look at Xeljanz, for example, they have a black box warning saying that you might get, it might cause gastrointestinal tears, for example, which is, of course, nothing you want when you already suffer from ulcerative colitis. Another way of looking at the medical need, but also the commercial potential, is to look at the licensing deals and M&A transactions that have been done within this space over the last couple of years. And there is a string of deals with very high amounts involved. So I've just made a selection here of the biggest ones of the last 4 years with Celgene that bought Receptos. It was an outright acquisition to get their hands on Ozanimod, which is an S1P receptor modulator that is also being developed for MS. They paid $7 billion for that. Then if we look more recently, the last one here, last year, when J&J licensed an asset from Theravance in California, it's a gut-restricted JAK inhibitor. They had just finished phase 1, and they got $100 million up front, plus almost $1 billion in potential milestones and royalty. And you can see that the upfronts have been between the 90 and 300 million in the last couple of deals here. So that's another way of showing the commercial potential in this indication. So this is where we come in with Cobitolimod, our lead drug candidate. It's a potential new medication for moderate to severe ulcerative colitis. So these are the more tough patients. They have typically failed a number of treatments previously, so they are treatment refractory. We have achieved clinical proof of concept in moderate to severe ulcerative colitis with a very favorable safety profile, which is one of our strong selling points here. We have a lot of clinical experience of this drug candidate. We've been through 4 clinical studies, and we have another one ongoing. 249 patients have received active drug, so we have quite a big safety database on this. So what stands out about Cobitolimod versus the competition is that we have a novel and unique mechanism of action. So as I showed previously, there are a couple of mechanisms of actions approved, and there are more on their way to market. And there are many versions or many drugs per mechanism of action. But we are alone with our TLR9 agonist mechanism of action, which will be important because not one class seems to help all patients, and even the patients that do respond initially to a class might develop tolerance to that, and they need to rotate. So it's important to have different classes of drugs that patients can rotate around. We have in clinical trials shown a higher efficacy than the currently approved biologics in an indirect comparison, with a very superior safety profile. It's a local treatment. We give this rectally, so from behind. It's a 50 milliliter solution that we give in an enema, which means we get straight to the site of inflammation. We give this straight to the inflamed colon, which one provides a rapid onset of action. We don't need to build up a systemic concentration to treat the gut. We actually go straight to the gut. But secondly, it probably explains the superior safety profile in that we don't have a lot of this drug floating around systemically, causing off-target effects. IP-wise, we are protected beyond 2030, and all in all, we see Cobitolimod as having a high market potential as a safer and more efficacious alternative to the currently approved biologics or JAK inhibitors. Shortly on the mechanism of action then. So as I said, this is a short snippet of DNA that we use to modulate the immune system via toll-like receptor 9 or TLR9. Toll-like receptor 9 is the innate immune system's pattern recognition receptor for microbial DNA. So this is something that we have developed over millions of years to know when we get invaded by bad bugs. So we have a lot of TLR9 in the skin, in the airways, and in the gut, typically where you would encounter microbes coming from the outside. And we've built this piece of DNA to mimic a piece of degraded microbial DNA. So it looks like a piece of bacterial DNA. And toll-like receptor 9 can then reacts to this then and modulates the immune system. And as you probably know, we have a lot of microbes in our gut that we collaborate with. So not all bugs are bad. I mean, we have as many bacteria in our gut as we have cells in our body. So you don't want all bugs to react to, you don't want to have an inflammatory response to all bugs. And that's what we use here. So depending on the sequence of this piece of DNA, we can steer the immune system towards a anti-inflammatory response or a pro-inflammatory, but in the case of Cobitolimod, we steer it towards an anti-inflammatory response. So by mimicking microbial DNA, we can steer the immune system to have an anti-inflammatory response locally in the gut, which leads to healing of the colonic mucosa then. So if we look at our most recent clinical study in moderate to severe ulcerative colitis with Cobitolimod, we had about 130 patients in the study. They had all moderate to severe active ulcerative colitis. They were in a flare. Despite being on a lot of other treatments, all of these patients were on steroids, they were taking immunosuppressants. 40% had tried biologics, but they were currently not on biologics, but they had failed biologics previously. So a typical tough, moderate to severe patient population. They received 2 doses of 30 milligrams of Cobitolimod at baseline, and then another dose at 4 weeks. This is just before the second dose, the data I'm showing here. And we saw, after 1 treatment, 4 weeks later, a significant difference in the symptoms between the actively treated patients and the placebo-treated patients. How to define remission or symptom-free has been an area of debate in this indication. Now, both FDA and EMA, the regulatory authorities in the US and Europe, have issued guidance on what they want to see as an endpoint in this study, how they define symptom-free. And it's 3 things. You shouldn't have any blood in your stool when you go to the toilet. You shouldn't see any blood. You should go to the toilet a normal number of times per day, or slightly more. And the colon, when you do a colonoscopy, looking at the colon or filming the colon, it should be healed or maybe mildly inflamed, but there should be no ulcers or wounds or bleeding in there. If you fulfill all these 3 criteria, then you're considered to be symptom-free. So we just count how many patients in each arm actually were symptom-free, and we compare them, and you see we have almost a 19% difference or delta between the groups with a statistically significant p-value in that indication, which is now the one that the regulators want to use. So if we look at this, we only gave 30 milligrams of drug twice. We think we might get even more efficacy if we give more drug. So we've seen competitive efficacy in the clinical trials with low and infrequent dosing. We've had an excellent safety profile, both in our clinical studies, in people, and in toxicology studies in various models. So there's room to increase both the dose strength and the dose frequency here. And we have both clinical and preclinical data suggesting there is a dose-dependent effect, meaning if you give more drug, you will have a better effect. So we then decided to do a dose optimization study where we increase both the dose and dose frequency, hoping to substantially increase the effect while maintaining the excellent safety profile. So this is the study that is currently ongoing. It's a phase 2b dose optimization study. We call it the CONDUCT study. It's a 5-arm study. 1 arm receives 30 milligrams twice over the course of a month, which is what we gave in that last study. 1 gets a 4 times higher dose, and 1 gets an 8 times higher dose, so 250 milligrams given twice over the course of a month. And 1 group receives 125 milligrams every week, so they have a higher frequency. And then we have a placebo arm. In total, 215 patients, or 43 patients per arm. We read the primary endpoint, the primary efficacy measure at week 6, which is blood in stool, number of stools, and endoscopic reading. And then there is a follow-up visit at week 10. So we had our first patient in June 2017. This study is being run at 90 clinics around Europe. We're in 12 different European countries, from Sweden to Spain, from France to Russia. We cover almost all of Europe here. We have more than 180 patients that have completed the study without any safety concerns, so the safety profile seems to hold. We do not know if it's efficacious because the study is blinded, double-blinded. The patient recruitment is expected to be completed by June. We have less than 10 patients left to recruit, and then 8 to 10 weeks after that, we will have the top-line results. So it's after the summer sometime we'll have the results, which will be very exciting. As I said, this is a rectal formulation. Currently, we have also built a prototype of an oral formulation, meaning a capsule that you eat and then survives the stomach and releases the drug in the colon. So it's again a topical treatment, although you take it orally. That would be a potential follow-on product to the rectal one and would let us go after other indications in the gastrointestinal tract that we can't reach with the enema formulation. For example, Crohn's disease, which is a sister disease of ulcerative colitis. I also mentioned that we have more than a broad portfolio of other DIMs similar oligonucleotides. We actually have more than 150 of those constructed. Some of them give an anti-inflammatory response, so they can be used in inflammatory diseases, and some give a pro-inflammatory response that could be used, for example, in cancer, immune-oncology. The concept of TLR9 modulation will be validated by Cobitolimod. So if Cobitolimod works, then we see a big potential in these other, at least some of these other DIMs. Financially, we don't make any money, which biotech companies usually don't. We spent about 20 million the first quarter of this year, and we spent about 20 million per quarter also last year. At the end of the first quarter, we had a bit more than 60 million of cash, so that should see us through 2019 and definitely through the results. So we can unblind this study without any issues. So, in summary, we are active in a disease where there is a large commercial potential. It's a debilitating disease with high unmet medical need. The currently approved biologics sells for more than $5 billion per year, so it's an established high-value market. We see Cobitolimod having a high potential as a safer, more efficacious alternative to the biologics with a novel and unique mechanism of action. We've come a long way. We are at late-stage clinical development. We have a lot of clinical experience. We've had a great safety profile. We are currently recruiting the last patients for a phase 2b study, and we expect readout of that in the third quarter this year, so in the coming months. The study is designed to provide a substantially higher efficacy than are currently approved drugs, which would make it an attractive asset for potential partners. And it will also validate the broad portfolio of other DIMS compounds that we have with potential in inflammation. And again, we have a very experienced board of management guiding this. And that's where I will stop then and open for questions.
Thank you very much, Peter. It's a horrible disease, I must say. But it's very interesting with this conduct study that will you, hopefully in June, you can tell us more. But what is your strategic thinking, whether the result will be good or slightly disappointed? What is the next step then?
So first of all, what is good and what is slightly disappointing, that remains to be seen. But I mean, if we have at least 10%, then we're at least equally good on the efficacy side as the biologics, but with a superior safety profile. And if we get towards 20%, then we would actually be better than anyone that is under development in this indication. But I think either way, we would look for some sort of partnership because taking the next step, which is phase 3, will be quite costly, and it would be good to have somebody share the financial burden of that and contribute other resources, for example, commercial expertise and planning. I think the data will tell us what sort of partnership that will be. But if we have outstanding data, my feeling is that Big Pharma will be more interested because they are looking for really new revolutionary things and they can pay for them and they pay good money for those. With slightly less good data, I think it might be more a play for a specialty pharma company. There are a number of specialty pharma gastrointestinal or GI companies out there that are looking for products that can't pay the money that Big Pharma pays, but they would still be a good partner and could turn this into a quite nice little product.
So I think that also explains the thing why you haven't been acquired yet. Is many are waiting for your conduct study. That's the best guess.
Yeah.
If you see on the M&A market in general.
Yeah.
I think so. Is there something else that you want to highlight apart from your core focus for the conduct study that you think are interesting for your company right now? Or is that the main focus currently?
Absolutely. I think the main focus now is to get those last patients in, prepare the data unblinding, so we are prepare our different work streams regarding business development, regulatory, to plan for the phase 3 and everything else. So there's a lot of preparations going on. But I also want to mention again the other DIMS assets that we have, that if Cobitolimod now works in this study, it will be a validation for the whole platform. And there we have some promising preclinical data in other inflammatory indications for a couple of these that we could build on to broaden the portfolio or the pipeline going forward.
Okay. Okay, thank you very much, Peter. Thank you.